MIT engineers have designed a two-component system that can be injected into the body and help form blood clots at the sites of internal injury. These materials, which mimic the way that the body naturally forms clots, could offer a way to keep people with severe internal injuries alive until they can reach a hospital. In a mouse model of internal injury, the researchers showed that these components, a nanoparticle and a polymer, performed significantly better than hemostatic nanoparticles that were developed earlier. Unlike previously developed hemostatic systems, the new MIT technology mimics the actions of both platelets, the cells that initiate blood clotting, and fibrinogen, a protein that helps form clots. Blood loss from traumatic events such as car crashes contributes to more than 2.5 million deaths per year worldwide. This kind of blunt trauma can cause internal bleeding from organs such as the liver, which is difficult to detect and treat. In such cases, it is critical to stop the bleeding as soon as possible until a patient can be transported to the hospital for further treatment. Finding ways to prevent internal bleeding have an especially significant impact in the armed services, where delayed treatment for internal hemorrhage is one of the largest causes of preventable death. When internal injuries occur, platelets are attracted to the site and initiate the blood clotting cascade, which eventually forms a sticky plug of platelets and clotting proteins, including fibrinogen. However, if patients are losing a lot of blood, they don't have enough platelets or fibrinogen to form clots. The MIT team wanted to create an artificial system that could help save people's lives by replacing both of those clotting components. To achieve that, the researchers created a system with two types of materials, a nanoparticle that recruits platelets and a polymer that mimics fibrinogen. To test the system, the researchers used a mouse model of internal injury. They found that after being injected into the body, the two-component system was highly effective at stopping bleeding, and it worked about twice as well as the targeting particle on its own. Another important advantage of the clots is that they don't degrade as fast as naturally occurring clots do. When patients lose a lot of blood, they are usually given saline intravenously to keep up their blood pressure. But this saline also dilutes the existing platelets and fibrinogen, leading to weaker clots and faster degradation. However, the artificial clots are not as susceptible to this kind of degradation. The researchers also found that their nanoparticles did not induce any significant immune reaction in the mice compared to a glucose control. They now plan to test the system in a larger animal model, working with researchers at Massachusetts General Hospital. In the longer term, the researchers also hope to explore the possibility of using portable imaging devices to visualize the injected nanoparticles after they have entered the body. This could help doctors or emergency medical responders quickly determine the site of internal bleeding, which currently can only be done at a hospital with MRI, ultrasound, or surgery.